a very important opportunity to hear someone that is a true specialist on this topic. The topic today may seem a bit hard to us, but it's a very important topic: electroporation from the laboratory technique to a technological platform, the complexity and interdisciplinary approach. But let me just address you with some very brief presentation about our guest speaker. Damijan Miklavcic is a professor at the Faculty of Electrical Engineering, University of Ljubljana. He is the world leading expert in the field of electroporation. In the past 30 years, he contributed to a better understanding of electroporation on membrane, cell, and tissue levels. And he has been involved in basic research demonstrating improved efficacy of selected cancer drugs at the preclinical level by electroporation, developing electrodes and devices and providing numerical modeling towards treatment planning. He is also developing DNA delivery to cells by electroporation for cell and gene-based therapies. So, of course, he's a very well-known researcher. According to Google Scholar, his works received more than 33,000 citations and an H index of 100. So we have la crème de la crème, as we usually say, of EUTOPIA University's top researchers, and Nova is very proud of being able to invite and have this invitation accepted by one very and excellent researcher as Damijan Miklavcic. So please, Damijan, the floor is yours.
Thank you very much.
Yeah. Right now, I think everyone on the other side, yes, João is telling us that is everything.
Now, the only thing is the microphone here, which is a little bit unstable because the battery is dying. I'm sorry. It's the battery. So we're starting with a new set of batteries. And hopefully this one will work.
You bet it's okay.
So on the other side, I see people are being connected, and we see they are hearing. So we need to take care of our audience here in Cairo as well. Yeah. So 1, 2, 3, 1, 2, 3. 1, 2, 3, 1, 2, 3. Oh, it's 5. And this was 1, 2, 3, 1, 2, 3. 1, 2, 3, 1, 2, 3. That was working before. When we tested it, it was working. Now it's not very much. Now it does. Now it does. So we don't move it. And so thank you for the nice introduction, and of course for inviting me. The title of the talk is complex. It sounds interdisciplinary, but it's all about electroporation, because that's the only thing I know. So now you're questioning yourself, why do I listen about electroporation? We are very picky when we are young, yeah, what we are going to study. Okay, so now we need to move on. We have another problem, which was nicely working before. Yeah. Yeah, but we'll okay, so now so we are very picky when we choose our. That does not seem to be the problem. Let's hope with a little bit of technical issues at the beginning, we've cleared all the technical issues, and then we don't have to worry about it. So at the beginning, we are very, very picky which subject we will choose to study, which subject we will do for our thesis, and so on, because we only see the expert knowledge. In this case, it's electroporation. I found this image on LinkedIn, and I think it's really it tells the essence of whatever you do. Really, in reality, you learn much more than just the topic that you are focusing on. So you train your curiosity. You do, of course, adapt to different languages. You read, and you increase the ability to learn, communicate, critical thinking, and many other things. Time management, of course, is one of the critical. Project management. We all do that, and we don't think about that in that way. So take this as a lecture about these skills rather than about electroporation. And at the end of the day, at the end of the talk, you might actually learn also something about electroporation. So electroporation actually did not exist as a word, as a term before 1982. It was coined by Professor Eberhard Neumann, a dear friend of mine. And he first used it in this paper, where they introduced genes into mouse glioma cells. And these cells started to express proteins which were not naturally expressed by these cells. The method was rapidly adopted by many researchers, particularly in working with bacteria in cells and producing now new DNA for research in labs. It was previously almost impossible to do it. Now, all of a sudden, became very simple. It revolutionized the work in the labs, creating new possibilities, new species. It also allowed actually to produce now new proteins, and we are still seeing the development of this today. You may remember, of course, the famous sheep Dolly. Well, you didn't know by now, but the electroporation had fingers there as well in cloning the mammal. And not only that, we are hearing about this incredible cell therapies based on CAR T cells, which is, of course, it's pretty amazing because we can now reprogram our patient's immune cells to attack tumors. And this is really something that is very interesting. And even though there is not a single word in this paper published in Nature showing this breakthrough technology, it actually appears in the text about 94, almost 100 times. And without electroporation, it couldn't really be done. Now, in the world, primarily in the States and Europe, there are 6 therapies since 2017 that are directed towards various hematological cancers that are being used. In this treatment, the patient's blood is removed to get patient's T cells, the immune cells. They are then modified and reintroduced into the patient, where actually now these cells that are modified, that we teach them how to recognize the tumor, are being very efficient. There's currently more than 700 clinical trials around the world trying to address not only hematological cancers, but also some autoimmune diseases and also some solid tumors. So there's a lot about, let me say, gene transfer going on in the world. And it's very, very interesting and very promising. While a set of researchers was working with electroporation in bacteria and working in biomedicine, there were other people in the world. I call that the parallel universe. They were working in trying to use, of course, electroporation for the extraction of juices from fruits, for canning, for sterilization without thermal sterilization, because with the thermal sterilization we lose the vitamins, and of course the food starts to look very different. To extract sugar from the sugar beets, and also, of course, by applying PEF in different various areas of food technology. These two worlds were actually very far apart. Now, I noticed that the French fry is very popular in this country. And the French fries actually nowadays, it's prepared industrially. And all of that French fries is actually electroporated. I'm absolutely sure you have no idea about that. So when you eat your French fries, this actually has been electroporated. What it does is it changes the structure. It becomes soft. It's easier to cut it. It's much nicer because it has clean cuts, as you can see here on the screen. It reduces the cutting force, the need for the cutting force. So the knives are longer usable, or you can actually use the stream jet, the water knives, as they are called. During the frying, there is also less oil being used, which is very important for McDonald's and these chains because they use massive oil numbers. So this is how you get a perfect French fry with the pre-treatment of electroporation. And it's actually, this is worldwide, and these are the huge electroporators. So other places or other topics, other uses in the food technology where they don't use the word electroporation because the consumer doesn't like it, but they use the PET treatment, which is shortening for pulsed electric fields. So it's a PET treatment. It's not that bad as electroporation. Yeah, you don't want to have electroporated juice, but PET treated juice is okay. Yeah, so we can use PET treatment to reduce the microbial burden, to reduce spoilage of the beverages, improving sensorial properties, reducing the use of preservatives, improving food functionality, reducing waste, optimizing process efficiency, like in French fries, and of course, improving nutritional properties. All of that can be used. Here is to teach you a little bit of Slovenian. So I'm sure you're fluent in Slovenian. If not, okay, let's switch back to English just to make sure. So the comparison of thermal pasteurization and electroporation or PET treatment, you see actually the control through the days. So the French orange juice is very good, all are equally good, but then you start to see how the separation occurs, and this is no longer good. By the day, I don't see actually the day, which day it is. Yeah, you don't see it neither. So let's move this away. Yeah, so by the day 28, the thermal juice is no longer good. The electroporated juice may still be okay. Now, these producers are commercialized and are used, and you can read on the label they are mildly pasteurized. Mildly pasteurized. They don't even use the PET. Okay. So these two universes never collapsed. They never met each other until I was invited to one conference long time ago in France by a colleague, Eugene Vorobyev, who invited me to give a talk on cancer treatment. And I said, but this is the food conference. Why do you want me to come? And he said, that's exactly why I want you to come. So based on that first, I would say, clash of the two worlds, we started to prepare, I suggested to prepare actually the cost action, which was approved and started to run in 2011 until 2016. And there was many people involved, many countries, and we organized a number of sessions, number of schools and workshops. And of course, we also facilitated the scientific exchange, which we were calling the short-term scientific missions. So, all in all, by the end of the project, 4-year project, 5, we had almost 600 people from 43 countries, 247 institutions, and 30 companies were involved. So that's where I think we really started to realize that we are doing the same thing to different subjects or different objects. We organized then the first World Congress, and we have now the 5th World Congress. Every time we have roughly 400 people from around the world. And of course, we formed a society, and you're not going to believe it, we have a newsletter every month, which goes to 1,500 addresses. We are now preparing the second handbook, and this is actually a handbook that was published in 2016, I think, 17, as a materialized book. And this is actually a 4-volume book. So it's almost 3,000 pages, all on electroporation. So if you can write 3,000 pages on electroporation, maybe there is something to it. Huh? So electroporation at work, we've talked about French fries, we've heard about the juices, and we've heard about the famous sheep dolly. What about wine? Well, the wine, at least now I come from wine producing region, and I think it's important to know that in addition to, let me say, structure improved by changing the structure of the French fries, we can also use PET treatment for easier peeling of tomatoes, for example, for microalgae, for protein and extraction of lipids, that is the oil. And of course, the wine can be also treated now, and it's approved for use in the production of wine. Cooking is very popular, and I can tell you that has changed the human, because that was the only way how the brain grows, so that we can now study without really being exhausted. Yeah? And I can tell you that the future of cooking will be by electroporation. Don't tell me when the future will come. It may be in 10 years or 20 years, but it will definitely come, because the food is cooked in seconds. And there's no waste, because how much you eat, that much you put in. You need more within a few seconds. You have additional food on your plate. So, what is electroporation? I'm going to come back to this question a couple of times. So, is it important? Is it relevant? Is it important? You know, if we look, it's used in medicine, in food, in bioprocessing. You can electroporate all kinds of cells, eukaryotes, prokaryotes, and archaea. Now, you look at the number of patents in the patent scope of the World Intellectual Property Organization, and you can find, maybe by now you can already find 500,000, but it's several hundreds of thousands of patents on electroporation. If you look at the Web of Science, there's roughly 1,000 papers published per year, and the number is growing. And actually, I had to cut down the number of papers that you can get and so on, because it's just the numbers were too big to allow me as a, let me say, non-professional user of the Web of Science. Now, science patents are usually actually filed by companies, because patent costs, the maintenance of patent costs, that means that the companies can afford it. We in academia, even if we come up with something very interesting, usually we don't have means to actually file a patent and maintain a patent. What's the purpose of maintaining a patent if you don't earn money? It's just spending money for lawyers, and that's not a clever idea. Now, some years ago, so actually 2 years ago, I came across an analysis. The market value for 2021 globally was estimated to be 1,000 million euros. Now that's 1 billion. Yeah. In North America, half of that. And this was before the boom that I will talk about. So these numbers contain the equipment and everything what is sold worldwide for electroporation. Yeah. So it might be interesting. And I'm going to focus from now on, not on food, yeah, but on medicine, on electroporation-based medical treatments. And particularly, what I will do is, of course, I will talk about tumor treatment, gene therapy and DNA vaccines, and treatment of cardiac arrhythmias. Now, the medical use of electroporation, what brought me in this field was really electrochemotherapy. So again, what's electrochemotherapy? Well, electrochemotherapy allows to treat cancer treatment more efficiently with the combination of the drug and electroporation. This is actually the drug which brought me into this field. I started at the World Cancer Congress as a PhD student in 1989 and decided this is cool, this is so important. Why is this so important? Do you see on the graph? A cytotoxic drug called bleomycin, which is used in cancer treatment with the use of 8 100 microsecond pulses, becomes 1,000 to 10,000 times more efficient, which means that you can either really eradicate tumor or you can use 1,000 times lower concentration of the drug. And the person here is Louis Mir, who I then decided to spend my postdoc with him in Paris at Gustave Roussy. So that's where I started, and it was very simple. I said, we need to bring this to the patients at the Institute of Oncology in Ljubljana. I was attending the World Cancer Congress with colleagues from the Institute of Oncology, where I was working in experimental oncology for my PhD. So the concept is simple. So not only it's efficient, it's also very easy to explain. So if you choose the right drug, and the drug has intracellular target, it may or may not reach the intended target. If the target is inside the cell, and that is usually the DNA, and the drug cannot enter because of the plasma membrane, because of the membrane of the cell. Remember, the membrane of the cell is there just to make sure that things don't go out and things don't go in. Yeah. Now you deliver 8 times 100 microsecond pulses, and all of a sudden, the drug can come in. The membrane reseals. You can select the parameters to survive, so the membrane survives, and that the drug is entrapped and now has all the time to kill the cell. So this is, instead of continuous infusion of the drug, it's a single injection of the drug. It's a single treatment by electroporation, and the tumor disappears. Okay? So it's not weeks of chemotherapy. It's one single injection, and then you deliver the pulses, and then you wait, and the tumor virtually fell off. So if this is so beautiful and so impressive, so why didn't you hear about it? Because I'm sure you didn't, yeah? It doesn't really have. So the comment was, maybe it has a drawbacks. Well, everything in your life has a drawback. There's no free lunch, I think, the English say, yeah? There's no free lunch, yeah? You will pay it at the end. Now, let's look at it. What I showed you is actually the mechanism of electrochemotherapy is a combination of electroporation and the drug. So, and I explained that the drug gets in because I electroporated the membrane. It's not only that, there are other, of course, effects that contribute to it. And we have been working with colleagues around the world and around Europe for decades. And of course, now we know much better. It's not only that the membrane is electroporated and the drug can come in. It's also entrapped because we stop the blood flow for a period of time. The drug, which gets into endothelial cells in neovasculature, the newly developed vasculature in the tumors will disrupt it. Plus, it will trigger the immune response. So all of that is really orchestrated for eradicating tumors locally. It's a local treatment. That's one of the critical issues. It's not a systemic disease. Cancer is a systemic disease. So the only part that is contributing is the stimulation of the immune system to the systemic treatment. So that's the drawback, one of the drawbacks. Now, how do we do the treatment? If the tumor is on the surface, it's very simple. We can either inject systemically the drug or intratumorally, as you see on the left photo. Then we bring in the electrodes and we deliver the pulses. And as I said, then we wait. Now, not all the tumors stay on the surface. So there are internal tumors. So for me as an engineer, my background is electrical engineering, biomedical engineering. I was immediately intrigued, why not treating deep-seated tumors? So metastasis in the liver, for example, metastasis in the brain and so on. That's usually what, people are learning about. And so treating deep-seated tumors, yes, you can do it. You can use, of course, needles, and interventional radiologists will place it. They will ask, how should I place them? Where should I place them? Yeah. You can have endoluminal treatments for esophagus or the colon. And that can be done, of course, with the use of special device that you put on the endoscope. Or you can treat with, let me say, less invasive electrodes. And we have been part of development of all of these electrodes and all of these treatments. Mainly to treat the deep-seated tumors, the prerequisites are pretty much the same. Only the problem is that when you have the electrodes and the tumor on the skin, it's easy to, because you have the visual feedback. And if you, by any chance, you miss it, you can just slightly move it, yeah, for half a millimeter or 1 centimeter. When you are treating deep-seated tumors like in liver or in spine, you have no vision. Well, most of us don't. So you need to have something like, I don't know, ultrasound or some sort of irradiation that allows you to visualize where you're going to place it. Or you need a navigation system. And once you're inside, you can't really move very much. You can go in, out, but you can't go left, right, and so on. So you need to know in advance where you will be placing. So you need patient-specific treatment plan and image guidance for electrodes. Now, that's easier said than done because you need, of course, images from each patient separately. You need to process that, you need to segment that, let us say, many people are doing, but then you need to develop a numerical model, where to place the electrodes, and eventually you need to execute the plan. So we've done that, we've shown that, and it's really easy with all the navigation system, with all the medical robots nowadays, and with all the imaging systems to do it technically. But it takes time. Remember, one of the critical things is to actually develop a numerical model that will tell you where to place the electrodes. So this is one example with a patient with 2 metastases in a bony structure. And you see here how it looks like. We coupled that with so we first did the treatment plan. In yellow, you see the tumor. In black, you see the dots and the lines. These are the electrodes, where to place them. And then we connected the navigation system with this. And, you don't see the tumor, but the needle aspiration biopsy after a single treatment, performed after several weeks, showed that there are no tumor cells. I actually know a couple of patients who have been treated in this way, and they are still fine, they are still alive. And this is years after the tumor was treated. And they are not disfigured by extensive surgeries and so on. So it's really pretty amazing. Yet you haven't heard about electrochemotherapy. So that's, again, it's still a question. So one of the problems, I will come to it later. During our journey, we came across an incredible paper where they described a single patient experience. They treated the patient with the metastasis in the spine, in the vertebra, actually, and they did the electrochemotherapy. Now, the interesting part is that these patients usually die within a year. Now, this patient follow-up was 4 years, the last follow-up I know of. The major problem with this kind of tumors is the pain associated with it. And the pain in this patient actually reduced from 10, which is maximum, to almost negligible, which is 2. VAS stands for visual analog scale. We reached out to these orthopedic surgeons. They work in Bologna, and of course they used the device that we were helping to develop for a company, IGEA, which is not far from Bologna. It's in Modena. And we said, we would like to really reconstruct that case. And they offered us what they had: the intraoperative images, the CT scans, the distances they measured, and of course the voltages and the current that they measured, the device measured. And we then, of course, we reconstructed. And what you see here on image A and B is already the reconstruction. Now, this treatment was quite invasive, and we were trying to figure out whether we can do less invasive treatment using approach which is called the spinal pedicular approach through the pedicles of the vertebra, which is the usual type of fixation for the orthopedic surgeons. And yes, you can do it. So nowadays, they start to use this in patients with this kind of metastasis, if nothing else, to reduce the pain, but also because other treatments are really damaging the bone, and the patients are compromised mechanically. So usually they cannot walk after that because their spine is too fragile. And so that's potentially one of the things. So remember the graph at the beginning when I said it's about electroporation, but you learn so much different things. Well, in order to bring the electrochemotherapy to the patients, we went to a large extent outside of our, how is it called, the confidence zone, yeah? So we had to reach out towards different disciplines. So we had to develop devices as engineers, that's okay, but it was high power electronics, and that's not always easy. You need to measure this, you need to process that online if possible, or during the treatment. Numerical modeling, we had to do everything from the scratch. This was back in the 90s. There was no console. The computers were poor, really poor. They couldn't really calculate a lot of things. So usually a tip of the finger was represented with 10 to 100 elements. Now you can place there 1 million or 10 millions, and you still do the computation easily. So that was one part. And of course, we needed to validate this kind of models. We needed to calculate not only electric field distribution, take into account how this changes during the delivery of high voltage pulses. We needed to support clinical trial. It was a multi-center study, and there was no electronic data collection system available. So we developed it with a software company who was developing the electronic collection system for car accidents at that time. It's pretty much the same, whether you do that for, clinical data collection or for the car accident collections. You have a form that you have to fill out. You have a photo that you have to attach, and that needs to be fed into the database. So whatever you do, you can use it in very different fields. And of course, we have to do the systematic reviews, meta-analysis, and stuff like that. So this is actually also a part of work that we didn't know before. So we have to learn. And at the end, being disappointed because it was not picked up, we didn't understand. And one of the drawbacks was the price, because the production is low, because the acceptance is not high. The prices are high, and the companies want to earn money back that they invested. So they always place the prices as high as they can, to be honest. Yeah. So they will sell as high as they can, even if that hinders a little bit the spread, of the treatment. So for that, we also did the health technology assessment, or as it's called, the early health technology assessment. And we published extensively on these different fields, really very different journals, very different fields. And for decades, I was receiving invitations by the pharmacy companies to come and present our work on electronic data collection systems. So this is not my thing. I'm not interested. I did it because I needed it. So, I'm not going to come lecture because I'm not the expert. Find somebody else. Yeah. It's really, you need to go to a distance. You need to go outside of your confidence zone. And you need to be very confident in doing that, because confidence zone, it's where you feel confident. You go out of the zone, all of a sudden, others know better. I don't know. No, they don't. There's nobody there. It's a desert. You need to reach out, because nobody's going to do it instead of you. You can pact, you can find the tribe that is doing something which comes close, and that is called interdisciplinarity. But you need to develop the language. They speak different language. Even if it's English, it's still different. Yeah. So what is electrochemotherapy right now? Well, we can treat metastasis and tumors on the surface, but that usually is not a serious problem. There are cases and there are patients who can benefit from that. In the meantime, as I said, many immune drugs came out, which were very efficient and are very efficient in treating melanoma. So melanoma is no longer a target for electrochemotherapy, to be honest. And the numbers were, not going as fast because there was a crisis in between 2008 and 2010. And of course, you see here, for example, this was a projection before the COVID. And of course, COVID also changed many things. The money goes different ways. Now, one of the issues, as I said, it's about electrochemotherapy not being systemic treatment. So that's the major drawback. That, and of course, the price. The pricing will change but this drawback will stay. So it's never really considered as prolonging the life of the patient, even if it does, but it's rather about improving the quality of life. So maybe we need to look at patients for whom this really makes a big difference. And one big difference is actually the spine metastasis. And most of the cancers, eventually, patients develop metastasis in spine. And that is extremely painful. If we can reduce that pain, if we can improve the quality of these kind of patients, I think this is a good target. There are other options, but this one is actually the best, I can tell. It will take time. Or we find an indication where there's no other treatment, like vascular malformations. And here, for example, it's a patient, you see actually a vein which is feeding this lip here, and there's no real treatment for that. So you do the electrochemotherapy, the patient after a single treatment, this is after 18 months or published by colleagues from the Institute of Oncology, it goes away. So that is something that potentially is a new indication. And that will help, of course, to spread the word of electrochemotherapy. So I spent 20 years in the field of electrochemotherapy, and I've become really disappointed because I felt this is such a splendid treatment and was not taken up. But I also became an expert. So there was a large company who reached out to me and they said, can we come and talk to you? We have an idea that we would like to develop, but we would like to pick your brain, as the Americans say. And so we started to work with them on something which is now called treatment of cardiac arrhythmias. Actually, it's called the atrial fibrillation treatment, and the company was Medtronic. So how come that this came around? So you can do an electroporation and the membrane, gets compromised, but the cell survives. This is important in gene therapy, and it's also an action of mode in electrochemotherapy, at least part of it. But if you deliver more pulses or more intense pulses, you can kill the cells on its own. You don't need the drug. And I got that question often enough. You can kill the drug, you can kill the tumor, you can kill the cells, you don't need the drug. I said, yes, but it's more clever. It's selective because the combination of the two and so on. They were listening, but not. Then a colleague of mine, Boris Rubinsky from Berkeley, said we can kill cells with electroporation in a non-thermal way. Wow. So what's new? Well, what is new is all the ablation modalities use thermal damage, freezing or heating. And that's very difficult to heal, and it's indiscriminate. And actually, the difference between the two, when it comes to ablating pulmonary veins, is actually the upper row, you see the narrowing of the veins going out of the left atrium. You see the damage to the lung, and you may not see, but there is a damage to phrenic nerve, which is close to the heart. With the pulsed field ablation, as they call it in the cardiac arena, so in the foot they use it as a PET treatment. Here they use the PFA treatment. Yeah? You don't see this kind of narrowing, you don't see damage to the lung, and you don't see damage to the nerve. So they believe it's really selective. So what is this cardiac ablation now? Why is this interesting? Well, I can tell you the market value for cancer ablation is about, I think, 300 million worldwide. For cardiac ablation, 1 billion. So the market size is huge in comparison to cancer ablation. Not only that, the number of people who are affected by cardiovascular diseases around the world is constantly increasing. So not only there are patients, there's going to be more and more patients around the world. And one of these, of course, the cause number one is cardiovascular. Only second is actually cancer. So also in this respect. And now cardiac arrhythmias with 60 million people is big because in Europe, in some countries, it only costs 3,000 per year to treat it. But in the States, it costs 63,000 US dollars. I don't know how much is in your country, but it's probably somewhere in between. Yeah. Now, the fact that this is interesting is also underlined by the number of companies, major companies, all the big companies of medical medtech companies are introducing and developing these devices. Medtronic, Boston Scientific, Biosense Webster of J&J, Apparent was a startup now bought by Medtronic. And 4 of these systems are already approved in Europe, and 2 are approved by FDA in the States. 1 actually Boston Scientific this year, Medtronic last year. So what is atrial fibrillation? So first of all, 1 out of 3, so you can count yourself here, 1 out of 3 or 5, 20 to 30% of you will have this kind of disease in a lifetime. Yeah, I'm sorry to say. I mean, there are other diseases that you can get. This one is treatable now with what we developed, so it's easy. It's better to have this one than something else that we haven't developed yet. So, atrial fibrillation, the numbers grow every year, and it's called of epidemic dimensions. And if not treated early enough, the quality of life is bad, but this is not the only thing. There's a risk of ischemic stroke, brain damage, heart failure, and it's also linked to dementia. So if you have this kind of disease, it's good to be treated, and it's good to be treated as soon as possible. The treatment can be either cardiac ablation or antiarrhythmic drugs. Antiarrhythmic drugs don't work. Yeah, I can tell you that. And cardiac ablation works, but it comes with significant problems. So, atrial fibrillation is actually the irregular beating of atrium, and they realized, or they identified, Michel Izaguirre and Piaget's and colleagues from Bordeaux, from France. They identified that most of the triggers are in the sleeves of the pulmonary veins. And then they suggested if we isolate these pulmonary veins so that these triggers do not send signals back to atrial, now this is treatment. The treatment is done. And the way how they do usually ablation is radiofrequency ablation. So one of the problems with this is, of course, we have different names for this atrial fibrillation, and whichever you have, the sooner the treatment is, the better. The problem is that very close, the anatomy of the heart is such that esophagus is very close by. It almost touches the left atrium. And there are also phrenic nerves and vagal nerves that are going nearby. So what happens if you are too aggressive in treatment is you can actually perforate, make the perforation between the esophagus and the heart. You don't see it, it develops. So the patient can go home and it develops later on. And there's no treatment for that. Now, there are safety concerns that, of course, need to be taken care of. As I said, the anatomy is preventing for really being following treatment because you're afraid that you're going to damage the esophagus and the phrenic nerve. So electrophysiologists, cardiologists who are performing this kind of treatment, they need to constantly balance between being efficient and really create durable lesion, or being safe, not risking too much the patient's life and the treatment. And as I said, the esophageal treatment, it's rare, but it happens. And when it happens, 2 out of 3 patients will die. So that's, that's preventing actually of treating simpler types of atrial fibrillation with this kind of treatment. That, and of course, it's costly. So they tried it with pharmacology, but pharmacology does not work, and then it gets worse, and eventually they try. It's still not life-threatening disease. And you go on a table and, 2-3 times out of 1,000, maybe, you will have this kind of complication. And if you are one of those, there's a big chance you're going to die. And you only have atrial fibrillation. So that's, should I go or should I stay? Yeah. Now, field ablation is ablation by electroporation. And it really started to grow. And the first time the pulse field ablation word appears in the literature is 2019. Not before. So there was no pulse field ablation before that. I think it was 2019 or 2020. So it's efficient. It allows to create durable, it shortens procedure time. Usually the procedure time is now about 2 to 3 hours for pulmonary vein ablation, which is tedious job for cardiologists. And of course, you need a dedicated studio for that, which is the EP lab, electrophysiology lab, and you need the whole team to support that. So it's expensive. Now, electroporation brings promise of being safe, and it proved to be safe. And when compared to historical data on efficiency and durability, it was really comparably efficient. And this was shown in 2 large studies, clinical studies. One sponsored by Medtronic, which was run in 40 different sites, included 300 patients, and the safety was superb. Never seen something like that in this kind of treatments. No esophageal events, no pulmonary vein stenosis, 0 phrenic nerve injury. So really the major clinical complications were absent. And if you compare this, of course, the efficacy is pretty much where radiofrequency and cryoablation, the 2 which are there, are working. But you have to know that this is now the first time used, and it's a first generation of devices in comparison to radiofrequency ablation, which has been perfected over 20-25 years to give similar results. So I can't even imagine where pulsed field ablation will be in 20 years. Yeah. The other study was actually comparing head to head, not with historical data, but really comparing head to head. Patients treated either by pulsed field ablation or treated by cryoablation radiofrequency ablation. Again, the treatment was pretty much similar, equal efficiency following 1 year. This was actually reported by Uwe Krabbe, published. They presented last year the results of a registry where they collected 17,000 patients treated so far with this treatment, and there was 0 esophageal events, 0 pulmonary vein stenosis, and 0 phrenic nerve paralysis. So this is something that is really, really looking good. But there are a couple of minor things that they did not expect. Of course, the treatment is different. So, are there any drawbacks? Well, as I said, there's nothing like free lunch. So, first of all, when you deliver the pulses, immediately the signals disappear. Now, before, cardiac electrophysiology depended on these signals. And they considered that once the signals are gone with radiofrequency ablation, they will not come back, so they can now move to the next point to ablate. Here, the first pulse you deliver, the signals disappear, and all of a sudden you are now blind in atrium, and you don't know where to place the next ablation point. So that's something that is not there. And it's disturbing because it changes the workflow. And you know, when you change the workflow, you are not comfortable. You want to do every day the same. You want to do to the same lecture hall, to the same restaurant. You know, it's just you don't want to change things. So now they will have to change. What they also see is other things. And I'll go slowly through. And of course, the big things that were the radiofrequency ablation and cryoablation are bringing, they are not on the table anymore, at least according to the data, clinical data and preclinical data available so far. But we need to look at other things. Are there other problems? Are there other, complications that they may arise? And of course, there's a lot of unknowns because this is new technology. And it was the first time presented in 2019. And again, the signals disappear almost immediately. And to be honest, at the beginning, they thought that's it. They didn't know it's going to come back. And I didn't know that they don't know that because we didn't speak the same language. Yeah. I didn't know that they are relying on this thing. So I started to discuss with them and I started to learn their language. And I think we have great things to achieve. And this is Atul Verma, with whom I spent quite some time, both in experiments, but I was also there for one of the first patients being treated in Canada. He's now at McGill in Montreal. Now, remember safety concerns, risk of phrenic injury, and I said there's none. And then they said, yes, but there is, but it goes away. Yes, of course, after a few minutes it goes away. So the signal is not conducted by a nerve, so that's not a big issue. It's known, and we can explain it. And actually, this was done in preclinical experiment, and it's very easy to explain. But at the chronic time, after a few weeks, there's absolutely no damage to the phrenic nerve. The function is back in patients and in animals. So why did it happen? Well, I'm going to show you that a little bit later. It's the game, it's the coincidence. But actually, before radiofrequency ablation was developed, there were people who were trying to use something what they call DC electrical ablation, and it's actually electroporation. But this was in 1982, when you remember the coin electroporation was only invented. And this was before the time of internet. This was before the time of electronic databases. So before you found another paper, before the paper was printed, it came to the library, and you went to the library and you dig through. I know a year was around, or 2 years, or even 3 years. Yeah. So it's more of a game of numbers. But what I didn't say, Eberhard Neumann, at the same time, when he used and coined the term electroporation, he also proposed a mechanism. And he proposed that in the lipid bilayer, there is a pore that is being formed, and now the DNA goes through this pore. I must say, and I must admit, that this is a very simplistic view of electroporation. And now we know, after 40 years of research, that this is far too easy, to be like this. So again, what is electroporation? At the end, you have a test. The question will be, what is electroporation? So the easy way is you expose a cell to the electric field, and because of the geometry and material properties, you get induced transmembrane voltage. Now, usually you have about 70 millivolts across the membrane in excitable cells like nerves and muscles. If this is much bigger, then what we observe is the increase of flux between the inside and outside, and it's indiscriminate. It goes either in or out. But then it can reseal, and the cell can survive. But it cannot, and then we call it reversible electroporation. It's important for gene transfer. But when we talk about just killing the cells, then the cells die, and we call it irreversible. As I said, it's a little bit more complicated because there's a sequence of events that actually electroporation triggers. So when you talk to different people, somebody will understand electroporation as forming the pores in the lipid bilayer. Somebody will talk about electroporation as the treatment, and somebody will use different words. So I think it's important to understand. You trigger a sequence of events which go from nano to microseconds and go out into days, hours for sure, 8 hours, 24 hours. And in vivo situation, in animal or in patient, it really goes into days and weeks. So I think this is important to understand. Let me just show you. We can test membrane permeabilization in cells in vitro. And this is the blue curve. And we can test the survival. This is the red curve. Now, the survival, if I subtract, I see it's different. I now call the percentage of cell death. There are two things that are important on this graph. And if you show it like this, is that all the cells that are being permeabilized are not going to die, which is good. But now the question is, if I want to kill the cells, how will I know now that my treatment is efficient and that the cell will die eventually? Well, I don't. And this is the part we are still working on, because now, with the pulsed field ablation in cardiology, this has become as a major question. Before, we were trying to avoid killing the cells, keeping them alive. Now, we need to understand why the cells die. And the two curves are not determined at the same time. I didn't say that at the beginning. So most of the time, actually, this is how it's presented. But these two curves are at least 24 hours apart, which means that we need to add additional dimension. That's why it's important to understand that there's a dynamics involved. Okay. So it's even worse. It's more complicated. As I said, there are no easy things. There are no simple explanations. There are simplifications, but there are no simple things. So you can achieve electroporation with any kind of pulses, nanosecond, microsecond, millisecond. Monophasic, biphasic, or monopolar, bipolar, exponentially decaying. And all of these will trigger a sequence of events which are virtually the same. There might be slight differences, we're working on them, but all of these pulses will deliver the same. It's not only lipid force, we find lipid oxidation as a consequence of force. We also see protein denaturation and partial unfolding of proteins. And all of that is in the membrane. And now, if we think about the waveforms, the pulse parameters that we can play with, we have at least that much. So we have 9 parameters that we can tweak. So it's the amplitude, one direction, the other direction, the plus, the minus, the duration, the repetition frequency, the delay between the pulses. Does it matter? Unfortunately, yes. So you need to optimize these 9 parameters. Where do I start? Yeah, maybe artificial intelligence will give us the answer on that one. But to show you really, I use the word artificial intelligence because somebody told me that I need to do that, to be contemporary, to be modern, to somehow introduce it into the talk. And that's my best attempt, okay? So I got your attention with the artificial intelligence. Now I'm also an expert on artificial intelligence. Now, does it matter? Yes. If we use the 8 times 100 microsecond pulses, sorry, this is 8 times 100 or 80, yeah, because it's the ablation. If I combine the 80 100 microsecond pulses, this would be altogether 8 milliseconds. And I determine the survival. You see the RE curve on the right-hand side graph, the length curve, is at about the 50%. Killing is at about 700 volts per centimeter. If I now use the same total duration of the pulse, but I cut it and make it 2,500 volts per centimeter to kill the same number of percentage of cells. Not only that, I use the same pulse duration, but I only change the interpulse delay between the positive and negative. It also makes difference. So the pause between the pulses makes difference. And whether I determine this at 3 hours or 24 hours, it also matters. If I move and go to 1 microsecond pulses, I see the same. So really, these pulse parameters are critical. And I said one of the key questions is about how to kill the cells or how the cells are dying, and knowing that would help us to predict the treatment outcome. And we started to look into that because we were not experts on the cell death. We started to look at the literature. And it's a mess. Some they say they die by apoptosis because of electroporation, they die by apoptosis. Some they say it's by necrosis, some they say it's by necroptosis, and some they say it's pyroptosis. So 4 different types of cell death. The other thing is, so which is it? Why are they different? Are they different? Yes, they are different. So then we started to look, what is causing the cell death? And we saw it's a membrane damage that is reported. We saw it's increase in ROS production, which are active species damaging then molecules around them, including lipids. Then a paper said DNA and protein damage, entry of calcium, mitochondrial damage, ATP depletion. So all of them. But what should I look if I want to predict? So the literature didn't tell us. So we needed to do experiments, and we are still doing experiments. But I'm a visual person, so I want to put things in. So now, instead of only having a lipid pore like presented in 1982, it's much more complex. You see many different things that we need to put in. Yeah. And when it comes to excitable cells, it starts to become even more complex because entry of the calcium can trigger contraction just because it enters in. And, you know, there are so many things going on, and I can't look at everything at the same time. And some of these are transient in nature. So when do I look at them? It's also very important. And they are linked. Yeah, the mitochondrial damage may be a consequence of calcium being introduced in high concentrations. And the DNA damage may be a consequence of something else. It's not the primary target. So what is really the primary target and why the cells are dying? So remember, this is only on the cell level. When I move to tissue and to patient, instead of cell membrane electroporation, I need to take care of the blood flow arrest, decreased vascular permeability, tissue porosity, eliciting immune response. All of that is there. And now, going into the heart, it's nerve excitation, muscle contraction, inducing pain and cough and stomach. So I need to understand all of that. Not one by one. Everything happens at the same time. Because when I talk about electric field, the electric field actually is distributed from the highest close to the electrode to the lowest farther away. Different electric fields of a given pulse duration will cause maybe irreversible electroporation, maybe reversible, a little bit further. Maybe there is going to be just the effect on the blood flow and tissue perfusion. And maybe it's only going to be stunning or nerve excitation and muscle contraction. But it's all at the same time. And how do these pulses affect different things? Well, in different ways. So the optimization, I'm going to use now, I had the opportunity to use the artificial intelligence again. So I'm now truly an expert on artificial intelligence. That this is really true is easy to demonstrate. You take a myocardium, cardiac cell, this one is from the right, and you pace it, and you see how it contracts under the microscope, and you determine how it contracts. You also measure the calcium at the same time, intracellular calcium, because these two are linked. And then you deliver the pulse, electroporation. And depending on the voltage or electric field to which you expose it, it will behave different. So it can actually stop. But after some time, when you start to pace it again, it will actually allow you to pace it again. So it's reversible. So that explains the disappearance of the signals, which then they come back. Knowing for many, many years that actually after electroporation, for a prolonged period of time, the conductivity increases of the membrane, which was measured by the patch clamp in cardiomyocytes, we decided to introduce that, and we added it to the Hodgkin-Huxley kind of model, which allows you to trigger and observe action potential by adding an additional conductivity. The switch actually here, the blue, is triggered, is turned on by the electroporation. And depending on how much how intense the electroporation is, how much the conductivity changes, you get actually a very different response. And we see all of these responses in our cardiomyocytes. So everybody wants to travel to see the new worlds. But the interesting part is that if you change the point of view and you ask your colleague to describe how the world looks like, you already are discovering the new world. So this is the real beauty of interdisciplinary work. And this is really something you shouldn't be afraid of, looking at things outside the box, they say. Yeah? Looking outside of the perimeter of where you feel comfortable. So all of that work couldn't be possible without the collaborators, without the lab, the members. We are right now about 25. Not everybody's in the picture, and some have left in the meantime, but nevertheless, a big thank you for them. Of course, it wouldn't be for the funding, because doing research is costly, and we were fortunate enough in the past years to actually acquire sufficient funding that we can do this interesting stuff. I would like to thank you with this for your patience, for your presence, and of course, this is maybe time. But if you are not done and you are not happy with traveling, like Marcel Proust said, you can still come. You can come to visit Slovenia, for example, either in the summer, for this conference, or maybe in November, where the weather is lousy, but you want to learn more about electrification. And that is unique worldwide course on electrification. Thank you.
Thank you. Now, let's open the discussion now. We may start with our vice rector, who is willing to address a couple of questions to our invitee, our guest speaker. So go ahead, João.
Yeah, well, thank you very much, Francisco. Thank you, Damijan. I want to first address your ability to convey this talk, because I think that what really comes out of this as a first important point is the importance and how difficult it is to do transdisciplinary research, and how sensitive the languages and the communication between different areas of knowledge and different fields are. I think that you gave an amazing example of how useful electric fields can be in various different areas of knowledge, being you coming from the Faculty of Engineering, Engineering, and moving into the food industry, into the health sector, biomedical industry. And I think that it's an amazing example of translating languages of different areas. I would address, I mean, your talk was quite extensive, so there are lots of different topics to be addressed depending on tastes. And I would address to three main questions that I noted out here. The first one being about the food transformation that you started with. So typically the food industry is considered bad boys in the sustainable environment, right? Most of the issues in sustainability nowadays are originated in, many of them are originated in the food industry. So I wonder, to start with, just a general question, how do you consider, if you could share with us, how those contributions that you referred to can help for a more sustainable environment? Because I think that there are a lot of things going on behind your presentation, and perhaps you'd like to share a couple of considerations on that point.
Yeah, thank you very much. So it's true. I mean, the topics that I tried to cover were more here to demonstrate how, you know, complex things are and how different things happen in different parts of the scientific disciplines. And it's really, it's incredible talking to people from different disciplines, even from food technology, to be honest. I learned so much. It's just incredible. And we can learn from each other so much that it's incredible. Talking about the PEF treatment, I mean, there are attempts in using and reducing the waste from food. So you can use the technology, the electroporation, to use parts of the food that have not been used now traditionally. Yeah, you can also facilitate the production, which means it's less energy consuming. So, for example, potato. Potato is a brilliant example of where, in order to cut it, because it's very hard. If anybody of you tried to cut a fresh potato, it cracks. It doesn't. It's very difficult to cut it actually. So, but if you cook it a little bit, blanching or something like that, then it becomes softer. But for that, you need to heat it up to at least 70 degrees for half an hour, and then you can cut it so it becomes soft. So now imagine the French fries, several tons per hour, tens of tons per hour in a factory. And then you need a tank to accommodate for that and to heat it up. So that would be, you know, tanks, I mean, like this room, you heat it up to 70 degrees for half an hour. It's enormous energy. But the other part is that this half an hour, it's a dead time for the production. So you need to have more of this in order for production to run, because all the rest is potato falls off the track, goes into the washing, and then goes into this tank. Then it's heated up, sits there for half an hour, and then it continues to flow to be cut, packaged, and so on. So now with electroporation, which is done on the flight, you don't need this tank. You don't need this energy. That's why it was taken up so easily, because the benefit is enormous. I mean, enormous. It's nothing in comparison to cardiac ablation, to be honest. But it's enormous in terms of not spending time, not spending the energy, and having a superior product. So I think there are going to be a lot of course, developments, and some are already there. But the companies are weighing very much, and they care about consumer acceptance, about the economics, about, you know, they will not introduce a new cooking device until the old ones are being used. And so when we talk about the cooking device, electroporation, for example, I've noticed here it's a gas. In Europe, mainly it's the inductive cookers, but not everybody has it. So they will wait until everybody has it around the world, and then they will introduce the new device. So it's, you know, it's economics. It's behind it. The world is ran by and dominated by earning money and profit and things like that. So it may take time. Sometimes we can take shortcuts, yeah, because it's really superb, like in potato, in French fries. But elsewhere, we will need more time.
Okay.
So yes, let me just follow up on that, because I don't know if Damon knows that. I'm a theoretical physicist, so I'm very keen on electromagnetic fields, and I'm impressed with your application as engineer of all those things. But I do also have an expertise in finance. I'm a professor of finance. So I do see the connection between the governance and the managerial constraints that the industry poses on the true development of things. That brings me to another thing. I'd like to connect that thing with health. I just had an ischemic accident a few months ago. One of the things the doctor told me is that I should avoid processed food. And it's a question in the back of our mind, how much this kind of PEF food is to be considered as processed food or not. And because in your presentation, you say it's very healthy, is it? Because then we do the transition to the health sector, right?
Absolutely. So yes, I mean, if you just imagine the reason why we cook, yeah, is twofold. One is to be able to get rid of the pathogenic bacteria and spoilage, so the yeast and the small little things that we don't see. So that is one. So to make it safe to eat. The other one is actually to make it digestible, because what is the food in the food is not the food that we eat, but it's the food mainly inside of the cells. So what we need to do is we need to release that to the outside. You know that there are animals who can eat raw food, yeah, and get things out, but their digestive system is completely different from ours. And you have some people who do not want to eat cooked food, so they only eat raw. And I haven't seen a single fat person of that kind. Usually they are thinner than they should be, yeah, because they can't get out of the raw food what they would need, actually, what their body would need. So that said, I think if we just think about not pasteurizing excessively to 90 degrees and so on, because all the vitamins are basically ruined, yeah, then this means that electroporation is the right answer. But of course, saying that, we have a lot of things that we work on, that we have to work on. And one of the things, at least in Europe, it's a big hurdle. I think it's appropriate to have, but it's the regulation of the food and of the new food in particular. And that, I think, is what some people and some companies are very much afraid of, because what is new food? The definition of the new food is newly processed food. Is it new food or it's not? So nobody dares to touch on that. So there's a directive which is called the New Food Directive, and of course that is a big unknown so far.
Thank you very much. I think your answer clarified actually number 2-3 different points that I had in my mind. Do you, Francisco, I'm allowed to do a third and final question regarding the health sector?
Yes, very briefly.
A final from me. I mean, I don't want to prevent other people to intervene, but my point is, being at a distance, I'm sort of eager to enter. That you see, for me, in the medical applications that you made, I find it quite interesting. But most medical evolution is dependent on pharma industry. A lot of those things, like treatment for AIDS, for COVID, more recently, right? All the vaccines, and we are strongly dependent on pharma, and that prevents universal access most of the times. So the treatments that you were describing, they seem to be much less drug dependent, but more technical, based on technical developments. That's and organizational involvement. Is that correct? Because what I see is that you have a number of hospitals and organizations engaged in your processes and trying and testing, and I don't see a name of a single pharmaceutical industry or things like that. It seems to be more. Please clarify me about that so I understand better a bit the process.
Yeah, I mean, the pharma is obviously it's always very interesting to know that a human, we are now used to take a pill and that will fix all. Yeah. Unfortunately, these pills don't always work. Yeah. So in cardiac arrhythmias, it was shown that cardiac ablation is superior to the pharma products. So that is one. The other thing is, of course, the Biosense Webster is a huge company which is a part of J&J. So these companies that I mentioned, the Medtronic, Boston Scientific, and so on, these are part of the med tech companies. And they are huge. Medtronic has 90,000 employees around the world alone. So these are huge companies. And of course, with the huge companies comes power to introduce new procedures, new treatments, new devices, and so on, because it's extremely costly. Yeah, maybe a little bit less than of course the pharma, but it's still very expensive. But let me just make a twist, and then I'm not going to dwell on this any further. Pharma is actually using electroporation in their products, so they depend on us. They don't necessarily know that.
Okay. Well, thank you very much. And congratulations for the talk. I thought it was very interesting and compelling. Francisco, I apologize for taking so much time, but...
No, thank you. Let's now extend the possibility of some of our colleagues from the auditorium here to address some questions. So I think, first, let's be very interdisciplinary. Our colleague is an economist, and she teaches marketing. So please.
So I can talk about there are no simplifications, but there are simplifications that will give you simplify things for me, someone from different backgrounds who understand the pleasure from them. So thank you so much. Very interesting and informative. And my question, I think, is a bit basic, but at the very beginning you said electroporation for natives, they named it treatment, and when it's put into Oregon, they have hospitalization. Are they the same things, but just different names or different terminologies? Probably people are not thinking of using the activated food, or are they different from one another?
Yeah, no, actually, the basic. The basic behind the PEF treatment and the PFA treatment and electroporation and electropermeabilization and nanoporation and nucleofaction and all of the other names that are being used in different fields is the same. It's the use of high voltage pulses to make the changes on the membrane level and to allow the transport of molecules to go across the membrane. Molecules that usually don't go, yeah? So in this respect, of course, the use of names come from two different reasons. One is the ego of researchers, because they need to do something new. And so doing something new, of course, is just giving a new name to your work. So I'm very much against that. I'm not saying my ego is not big. The other thing is, of course, that we have real studies in food industry, for example, whether the consumers would be willing to use electro something food or drinking electro something juices, and the answer was no. So it was a real, and these were big multinational companies involved. So they started to use the PEF treatment. And in partially PFA was introduced also based on this kind of thinking. We want to have, but it came from the company, Medtronic. They wanted to have a brand name. And so they came up with this pulse field ablation. I don't know how much was the marketing involved and so on. But it's very important how you present and what you present and the level of details that you present. It's also very important. So marketing is huge. I'm part of their campaign. Part of their campaign. They have invited me once there, and they were shooting me the whole day. Now they're cutting pieces and showing that even though it's 2-3 years old, but it was intended for, introducing this new treatment into the market and among the medical doctors.
Another very important question. When you said new food, this is something else from genetically modified food, right?
No, it's not. It's actually much broader. It's much broader. It covers all food that has not been on the market at that time when the directive was. So it's not only GMO, yeah, but it's many other things. Yeah. I'm not the right person, to be honest. We worked together. I ran that project. I learned a lot, but I didn't learn that much. Yeah.
Thank you so much.
You're welcome. Thanks.
Okay, it's Asma. Tens que ligar, é eu que posso ligar.
Okay, thank you so much for everything. I learned a lot. But I first have a question. I think it's kind of biology. How do you destroy the cell, the membrane that keeps everything in, just to put everything in without getting out? Answer for the question.
Yeah. So it's really, you can't do it so that you go in, but nothing would go out. So that's the level of detail that usually people tend to, not discuss. So they say we introduce genes, or they say we introduce drugs. Yes, but at the same time, things go out, things like ATP. And the ATP going out causes two things. It depletes ATP, so adenosine phosphate, which is actually the energy that the cell has for everything, to do everything. And so that goes out, leaking out, will cause a depletion of ATP. That can be a problem and can actually tell the cells that it's time to die. Yeah. But even if it does not die, ATP in the tissue is a signal. It's a molecule that now represents a signal for the cell has been damaged. This is the damage now. The macrophages need to come in and other immune cells to clear the damage. So you will have the inflammation. So this will be that's called inflammation, and of course, that of course tells you already that you can't have one or the other. You have a package, and if you want to really optimize something, you need to have all the inputs. You need to know the system, and you don't look at only one output because you need to look at multiple outputs. Multiple outputs that are relevant for you. So I want to maximize this. I want to minimize this. I don't care about this one, but I need to monitor this one because if it would change, then this will cause this and this. So that is, I mean, for me as a researcher, it's endless story, which means I don't need to change my field every 10 years. It's just going on and on and on. Just.
So that's the reason you did not mention leukemia, right? Because leukemia is all over the world.
And so leukemia is problematic because it's dispersed. Yeah? That's also why the CAR T, the cell-based therapy, is the most efficient and first developed for the other drugs as well. Were first developed for hematological cancers because these cancer cells are so easily accessible to drugs and these genetically modified cells. When you go to solid tumors, you deal with different. The drug does not get in, the distribution is difficult and so on. So usually it's just the surgery, or if the surgery is not possible, is radiotherapy. Yeah, but none of these two is really perfect. It should be avoided if at all possible. Yeah. You're welcome.
Yeah, Sophia wants to address.
About the AI?
No, no, no.
Sorry, this is a private joke. Yeah.
We already have private jokes in this life. No, it comes also what you just said. I don't change field. I can work on something new every 10th year. And I was wondering, because first you start with food, and then you start with killing a cell, and now you were talking about the arrhythmia in the heart. So what next, basically? Because it seems it has so many different applications. And first, how do you find a new application? Can we use it? Of course, I'm thinking, being an environmental engineer, in treating some type of pollutions, or how do we find something else to use it?
Yeah. So
to keep you in job, of course.
Yeah. So first of all, I really never worked in the food. I presented it, of course, as a parallel universe. What I did is actually I brought these two universes together. Yeah. So most of my work was actually in cancer treatment. And this was, that is the drug delivery. That is what brought me in. Yeah. And I thought at that time it's really simple. So we only need to bring it to the patient. Then it turned out we need to develop the device, we need to develop understanding, the electrodes for that. We need to do the numerical modeling, we need to do more experiments, we need to test other drugs and so on. So, and as we were doing this, others have found, not me, others have started to try to do, but since I somehow established myself as an expert, then they came to me and they said, what do you think about that? And, when I saw actually irreversible electroporation, and I saw in 2007 a paper by Jacob Levinsky and Boris Rubinsky, I said, and this I didn't mention, this is a clever way of using irreversible electroporation as an ablation. And they showed that you can actually ablate cardiac tissue. And it was since 2007 that I was waiting for a call because I couldn't persuade a cardiologist in Ljubljana to start working on that. So I was waiting for a call, and I received a call from a person from Electronic, Mark Stewart, and then we started to work with this company. It's been now since 2015, almost 10 years. So I must say that I'm super happy. I haven't done a lot. Definitely didn't invent anything, didn't come up with anything clever, but I was lucky enough to be part of development of electrochemotherapy. Just, as the train started to take off the train station, the first one, I jumped on. And the same was actually with the cardiac ablation. And when you ask me what's the next thing, well, first of all, electroporation is going to be everywhere. When people ask me, it's difficult to say where it's not going to be. Yeah, really, it's difficult because it's such a powerful and indiscriminate technique. But if you ask me, where's the next thing? It's the gene transfer, it's the gene therapies. Because just think, 2000 was the breaking point, not because of the 2K virus, but the human genome sequencing. But what was really done at that time was, it's like here in Egypt, until they found this triple plate with 3 different languages and 3 different signs, they couldn't decipher what the hieroglyphs really say. So we were putting down some sort of A, B, G, C, D, and so on, some letters that we couldn't really understand what they mean. And for most of them, we still don't. So the era of gene therapies is still ahead of us. And COVID showed one thing. Yes, we can do it. what was wrong was that they used the viral vectors to introduce DNA as a vaccine into the cells, because it's the viral vectors that cause the harm. So if instead of that you use electroporation, you're done, you're safe. Huh? And this is what we are doing in our lab for the past 10 years, basically. We are working quietly, very diligently on DNA. Not about drugs, not about treatments, diseases, indications. That's not ours. We're not the ones. We need to make sure that we understand how the DNA gets in, how the DNA gets expressed, and how to optimize that. And then move that to the level of tissue, which is far more complicated. And so and of course, developing devices and all of that, and the models and stuff. And then once this is done, it's the package. It's a brilliant package, and I think it's going to be the next big thing. That for me is the next big thing.
Thank you, Damijan. Okay, let's conclude this very interesting conference and discussion. Let's also express our gratitude to João, who is following us, and we will have the third EUTOPIA Cairo talk on the 10th of March. So let's applause Professor Damijan.
Thank you.
Thank you. Thank you. Bye, João.